Millions of animal and plant species of varying sizes and shapes inhabit the earth, and they all have one thing in common. They are all made up of cells, the fundamental unit of life in living organisms. Scientists didn't know about the existence of cells until the invention of the microscope. Anton van Leeuwenhoek, an amateur Dutch scientist, is credited as the first person to see and describe living cells. Later, Robert Brown expanded on Leeuwenhoek's studies and pointed to the presence of a cell nucleus. In 1838, German botanist Matthias Schleiden examined numerous plant species and concluded that plants have different types of cells which form plant tissues. A year later, Theodore Schwann, a British zoologist, examined different animal cells and discovered that the cells have a thin outer layer, which we now know as the cell or plasma membrane. He also studied plant cells and concluded that in addition to a cell membrane, plant cells also have a cell wall, a characteristic unique to plants. Schleiden and Schwann together formulated the cell theory, which stated that both plants and animals are made up of cells and their products such as proteins and lipids. However, the cell theory did not explain the formation of new cells, which was clarified by Rudolf Virchow in 1855. He coined the Latin phrase omnis cellula e cellula, which meant that new cells arise from pre-existing cells, and accordingly modified the cell theory, which now states that all living organisms are composed of cells and products of cells, and these cells arise from pre-existing cells. Cell research received a further boost with the invention of the electron microscope, as it enabled scientists to observe the structure of the cell. They observed that the structure of a cell varies in plants and animals. A plant cell, for instance, is surrounded by a distinct cell wall on the outside and a plasma membrane on the inside. Whereas an animal cell lacks a cell wall and has a plasma membrane instead that forms the outer cell boundary. Both animal and plant cells have a dense and spherical structure called the nucleus, which contains thread-like structures called chromosomes. Each chromosome is made of deoxyribonucleic acid or DNA, which stores the genetic information of the organism. If the nucleus is membrane-bound, then the cell is called an eukaryotic cell. They are found in members of kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. On the other hand, cells without a membrane-bound nucleus are called prokaryotic cells, and members of kingdom Monera possess such cells. Both eukaryotic and prokaryotic cells are filled with cytoplasm, a semi-fluid matrix present inner to the cell membrane. However, in an eukaryotic cell, the cytoplasm does not include the nucleoplasm, the nucleus matrix filled with the contents of the nucleus. Cytoplasm is a hub of activities and chemical reactions, such as protein synthesis, that help the cell remain alive. Suspended inside the cytoplasm are distinct structures called organelles. In eukaryotic cells, we find membrane-bound organelles such as the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies and vacuoles, as well as non-membrane-bound organelles such as ribosomes. Interestingly, ribosomes are also found in chloroplasts of plant cells as well as the mitochondria and rough endoplasmic reticulum of both plants and animals. Moreover, animal cells possess another non-membrane-bound organelle called the centriole, which aids in cell division. The cytoplasm of prokaryotic cells, on the other hand, lacks membrane-bound organelles but contains ribosomes and fragments of extrachromosomal DNA called plasmids. Cells usually differ in size and shape. For instance, mycoplasma measure only 0.3 microns in length, whereas human red blood cells are about 7 microns in diameter and nerve cells measure around 4 to 100 microns. Amazingly, the ostrich egg is the largest isolated single cell, and it measures 17 to 19 centimeters in length and 14 to 15 centimeters in width. Moreover, the shapes of cells range from disc-like to thread-like and from polygonal to columnar and cuboid. Today, though we have gained a lot of information about cells, scientists are still engaged in unlocking the secrets of the cell and its organelles. Millions of microorganisms are present in every nook and corner of our surroundings. Of these, certain microorganisms, such as bacteria, have a nucleoid but no membrane-bound organelles in their cytoplasm. Such organisms are called prokaryotes. Prokaryotes are smaller and multiply more rapidly than eukaryotes. All members of kingdom Monera, including bacteria, blue-green algae, and pleuropneumonia-like organisms or mycoplasma, are prokaryotes. And they can be rod-shaped, spherical, spiral-shaped, or comma-shaped. Microscopic studies reveal that most prokaryotes, except for a few such as mycoplasma and thermoplasma, possess a cell envelope, a complex, tightly bound, three-layered chemical structure consisting of the outermost glycocalyx, followed by the cell wall and then the plasma membrane. The layers together act as a single protective unit, though each has its distinct function. The glycocalyx, for instance, helps prokaryotes such as bacteria to resist antibiotics when they invade host cells. Did you know that the glycocalyx differs in thickness and composition among different prokaryotes? In some, it occurs as a thick and tough sheath called a capsule, while in others it may be a loose sheath called a slime layer. The glycocalyx is followed by a cell wall, which determines the shape of the cell and provides structural support, thereby preventing the cell from collapsing or bursting. Interestingly, the cell wall structure differs in different prokaryotes. In certain prokaryotes, such as Pseudomonas, the cell wall is composed of a thin inner layer of peptidoglycan and an outer membrane of lipopolysaccharide and lipoprotein. When such prokaryotes are subjected to the Gram staining procedure, they do not take up the Gram stain, but appear red or pink when counterstains such as safranine are added. Hence, they are called Gram-negative bacteria. On the other hand, the cell wall of prokaryotes such as Bacillus anthracis, composed of a dense layer of peptidoglycan and teichoic acids, turns violet when subjected to the Gram staining procedure. Hence, such prokaryotes are called Gram-positive. The cell wall is followed by the semi-permeable plasma membrane, which controls the entry and exit of small molecules like food particles and ions like chloride, sodium, and bicarbonates. Often, the plasma membrane extends into the cell, forming the mesosome